If you've been following me for a while, you might know that I'm really into methods that let you record biological information over time. And that's because the time-resolved nature of a cell is incredibly informative for understanding how biology actually unfolds. Organismal development, stress responses, drug resistance, everything that develops over time. The problem is that most ways we measure cellular information, like RNA, destroys the cell, precluding later sampling. But this new paper from Fei Chen's lab introduces a way to store cellular histories for later reading called TimeVault. They hijack these large, weird protein structures that already exist in human cells, called vault particles, and repurpose them as storage capsules for RNA. To make that possible, they simply fuse an RNA-grabbing protein to a vault protein. And they make it so that these vaults are formed inducibly, just add antibiotic, such that RNAs can be packaged into vaults at a time of the experimenter's choosing. Once inside the vault, those RNAs are protected for potentially weeks, and they're passed on to daughter cells and remain available for later sequencing so that you can reconstruct what the cell was expressing in the past. They show that this works transcriptome-wide with surprisingly little bias and minimal perturbation to the cell. And in a nice application, they use TimeVault to look at the histories of treatment-resistant cancer cells. And here they show that the cancer cells that managed to resist the treatment were already in a very particular sort of probably pro-resistance cell state before the treatment was even applied, which is something that would be very hard to pick up on with single time points alone. And while there are other comparable RNA recording methods that I also love, I'm especially excited about this one's compatibility with single-cell RNA sequencing, which is what will make this type of method really powerful when you can directly link individual cells' molecular history to its future fate. So I will be keeping an eye out for that inevitable future paper, and I'll be sure to keep you posted.